Hello everyone, my name is Trui Luyckx and I am a PhD candidate at the Laboratory of Food Chemistry and Human Nutrition at the University of Ghent. In the PROFIBFUN project, we investigated the propensity of food proteins to form amyloid-like fibrils. The other videos already showed some very promising findings and possible applications of food-derived amyloid-like fibrils, but we still have a very important question. What happens in our body when we consume amyloid-like fibrils? Well, to answer this question, we simulated the digestive process from mouth to small intestine, including all relevant enzymes. For ovalbumin, the most abundant egg white protein, this resulted in an incomplete breakdown of the fibrillar morphology. This means that amyloid structures reach the intestine and are available for absorption and transport to the rest of the human body. Intestinal cell models allowed us to evaluate the level of this transport by mimicking different intestinal cell types. Amyloid toxicity is mainly investigated in its medical context, where amyloids of certain human proteins are known to cause neural damage and lead to plaque deposition in neurodegenerative disorders like, for example, Alzheimer's and Parkinson's disease. The theoretical influence of food-derived amyloid-like fibrils on the general health status in both healthy and predisposed individuals was recently published in our review by Luyckx and co-workers. In practice, toxicity assays with ovalbumin fibrils as substrate ruled out direct cytotoxic effects on different cell lines, but there are also indirect effects that we need to consider. For example, can food-derived amyloid consumption cause cross-seeding inside the human body? The process of inducing amyloid formation from one protein through contact with preformed amyloid fibrils from another protein is called cross-seeding. To investigate whether or not ovalbumin fibrils can cross-seed cellular proteins related to, for example, Parkinson's disease, ovalbumin fibrils were brought into contact with cells that express the protein that is responsible for amyloid plaque formation in Parkinson's. Preliminary results showed that no cross-seeding occurred in the followed setup. Now it is time for some conclusions. In these videos, we have taken you to the exciting world of amyloid fibrils in food products. We showed that they are present in common boiled hen egg and that wheat gluten can also form protein fibrils under heating conditions that are relevant for food processing. Even more, protein fibrils from egg white proteins prepared under food-relevant conditions can stabilize emulsions, such as those found in mayonnaise. We have also investigated their formation at the molecular level, as we believe by looking into this we can design functional amyloid fibrils that can be used in food products. And as I mentioned before, I investigated their digestion in the human tract. The health effects of these protein fibrils in the human body will be further investigated. PROFIBFUN is an example of how interdisciplinary research can have relevant impact on designing food protein fibrils with potential innovative applications that can help us obtain healthy, sustainable, and tasteful food products.